Hello, hello, hello.
Hello, how is everyone doing? All one of us, all
Yes,
two of us.
yeah, exactly. Welcome to our first stream. We are,
We are not showing our faces yet, but we are now.
yes, good, because now everyone can appreciate the lovely message that I've written on the whiteboard behind us, because we are Science Scribbler, and this is our first QA and classify. I forgot what it said for a second.
Q&A and classify. And so today we're going to be talking a bit about the science Scribbler projects we have ish, maybe the origin story, and picking a project on the Zooniverse to classify
Yes,
with
while we are chatting.
while we are chatting
Yes.
about science
Mm-hmm.
and citizen science.
Yes.
Perfect. Okay. Do you want to
Share screen?
share screen?
Yeah, let's
Yep.
do it.
Okie dokie. There we go.
Beautiful. Lovely transition. So smooth.
Fantastic. I'm so good at this.
It's not our first time or anything. Yeah.
No.
Okay. Well, what are we interested in today? So this is the Zooniverse platform, which is one of the world's largest citizen science platforms. It's open to anyone. You literally just go to zooniverse.org, and you would have access to all of these different projects. They span a lot of different disciplines. I don't know if we have anything specific in mind today.
I suppose we can explore something new, can't we? I
Yeah.
mean, there is always the classic of Penguin Watch if we want to pick penguins.
Penguin Watch is a good one.
Yeah.
Yeah.
I
Okay.
wonder if it has data at the moment, actually.
Oh yeah. Let's type it in and see, because penguins are always fun. Oh, I don't think it does.
Oh,
Oh, there it is.
perhaps.
There it is. Penguins from above. That one's fun too, I guess. New workflow alert. Okay, so good timing for that. Okay, Magellanic or rockhopper.
I assume that's in reference to the type of penguin,
I would
right?
assume too. I know
Um,
rockhopper. I don't know, as you could
yeah.
probably tell, Magellanic.
I think I've seen rock hoppers before too, so I guess we could pick that one.
Okay.
Have a look at some penguins.
Yeah.
All right, so the first thing that comes up when you have a new when you're looking at something for the first time on the Zooniverse is you get a little introduction and tutorial. So yeah, I guess we can read through
Mm-hmm.
and figure out exactly how we're going to be contributing to the Penguin Watch's science research.
Yeah. Okay. So it looks like we are wanting to place marks on different rockhopper penguin adults, chicks, and eggs in the image. And you want to do that by clicking the center of each one's visible area. Oh, that's a fun term. And you can move the marks around to recenter them, remove any accidentals, or hide previous marks if need to. Okay, and we can learn more about what the penguins and chicks look like in the field guide. Oh, okay, yeah, so too small to count, so ignore any penguins that are too far in the background, like in the picture above. And you can click there were too many to count. Good. If it's too difficult to see any animals in the image, you can select too dark or blurry. Okay, good. Those are definitely not penguins. And we can also mark other animals if they're in the image. Okay, and those are all just different types of birds.
I guess the likelihood of seeing something else is not very high in these images.
Which is fair.
But there has been some safari style projects where you do often end up seeing signs of human life in some of the
Oh,
images.
yeah, I do remember that.
Yeah.
Yeah, yeah. There's like so I guess it's worth saying like most of these images from this style of project come from remote camera traps, right?
Yeah, triggered
Uh,
by movement
Triggered by
usually.
movement.
Or
So,
time series. Like we might be looking at time series data for
yep.
this one.
All right, I don't see anything in this image, so I'm going to go ahead and click next. You marked all the animals, yes. Done. All right. Oh, okay, now we've got more. All right. Oh, actually, before we continue on, let's check out the field guide because I don't know what chicks look like.
So we're going rock hopper.
Rockhopper, yeah. Okay. Smallest penguin species.
Picked an easy one. Absolutely.
Usually lay 2 eggs. Nests are dug into the ground. Chick or adult? Okay, here we go. Adults have got distinct yellow whiskers on their sides, red beaks and eyes. Their backs are black and their stomachs are white. And chicks have got, oop, I've clicked a button.
We can go back.
Let's try that again. Nope, I went the wrong way. All right, get back into that. There we go. Okay, field guide, rockhopper. That wasn't too bad. Okay, and chicks have got dark beaks, dark brown heads and backs, and cream stomachs. No head whiskers. Okay.
Yeah.
So yellow is an adult with red beaks. Yellow whiskers, red beaks, and eyes. And chicks are more browns with cream stomachs.
You can often, I think, also see that the chicks can be going through like that transitional stage of growth where they have like the fluffy brown bits coming off, right?
Yeah. Okay, fair enough. So we have adult with eggs. So we've got our red beak and some yellow whiskers. Adult with a young chick underneath. Oh, that is difficult to see. Okay. With an older chick, which makes sense. No red beak, no yellow. And then adult and juvenile. So again, no red beak and no yellow. Okay, so we're looking for beaks and yellow. Got it. That was helpful. Okay, and we can zoom in, as we're saying. So if we want to try to capture some of the smaller things, and we can pan around and drag the image. So let's start putting some points down, I think.
I think this was the first project I looked at when I went on the Zooniverse for the first time.
Yeah.
One of the first few that I had a look at, yeah. And I guess one of the things that I found immensely satisfying was finishing an image. Because sometimes you get empty ones, right?
Yeah.
Which are, I guess, not super interesting to look at. And sometimes you get ones which have so many penguins in the background that they're a little bit overwhelming.
Yeah.
But there's that like happy middle ground where you've got a couple, like, I don't know, 20 to 30, and it feels really satisfying to finish it off.
So I'm second guessing myself immediately because I've marked all of these as adults, but I can't actually tell from their markings. So I'm thinking I might need to delete these marks.
Do you think that they're too far away to be able to correctly identify?
So let's delete. And we'll keep these ones in the background as too far away. It's worth saying that for all of these projects, you are not the only person looking at the image. It's being looked at by many different people, so that, you know, different opinions can be taken into consideration, and ideally consensus is being reached on each image, so that you know, even if one or two people disagree, something will emerge that makes it more clear. Do you think that's reddish on that beak?
I think it's probably like sufficiently red, yeah. And I think that the one next to it has the illusion of whiskers, the whiskers that you can see.
Yeah, good. So we're on firmer ground here. I don't see whiskers on this one, so I'm going to call it a chick. Ooh, this one's doing a fun stance. It's like hitching a ride or something.
It's not the April Fools. Did you watch the April Fools video they had once where it was David Attenborough doing like a mockumentary about penguins flying into the tropics for the winter.
No, I don't think I've seen that.
I think it's probably a decade old now.
Yeah.
But it was a BBC April Fool's. So I don't know if you know the spaghetti tree one.
I don't know any of these. I'm not from the UK, clearly.
The BBC did an April Fool's joke by video, and again, it was a mockumentary. And it was in black and white, so I think it's pretty old. And they showed people harvesting spaghetti from the spaghetti trees.
Okay.
That was the whole pic, and people genuinely believed that spaghetti grew on trees at that point. Stephen Fry's done one where he was looking for the whale shark, and it was done like a nature documentary kind of thing, which was also good.
Yeah, that's fun.
Yeah, but the penguin one was quite funny. But second to that, I think, is another kind of CGI penguin gif that I've seen of one reaching out and slapping another as it falls down. I don't know if you've seen that.
No. You and I inhabit different parts of the internet. Okay, I think I'm gonna mark this one and then be done, because the rest of them I think are a bit too far away for me to see. So we're gonna click next. There were too many to count. Done. Okay.
This I classify as a satisfying image.
Yeah.
What do you think?
It's a lot sharper.
Yeah.
That's for sure.
Yeah.
Yeah.
Easier to identify, not too overwhelming.
Yeah. Smaller number.
Yeah.
Yep. Yeah. I still worry a little bit about those ones in the back as to whether or not we'll be able to tell if they are adults or not. Let's start with this one here. I think that's a red beak, so we're going to make that an adult. And let's pan over. Yeah, I think they're a bit too blurry for me to tell if they're adults or not.
I actually think, from my perspective, these two that are crossing over in the center look like they have a bit of brown, like fur, juvenile fur, still on their backs.
Okay.
And so I would classify them as chicks.
Okay. And so you would put the others as adults because they don't. Maybe that one also is.
Yeah, I think that one might be juvenile, and then I would err towards adults for the others.
Yeah. Okay. You've convinced me.
Well, this is another part, right, of citizen science,
Yeah.
like you say, with the consensus, that lots of people will look at the same images and they might disagree. And in this case, we don't know exactly what the right answer is. And what we're both looking at in the image might be different.
Oh, dear.
Or what we might interpret. That's pretty overwhelming. But the majority consensus is the thing that that comes out as being pretty good. So, you know, you ask me, you ask 20 other people, collectively we'll get it right.
Yeah.
Maybe I won't get it right every time.
Yes, yeah, exactly. Okay, we have to zoom in for this and treat it as quadrants, I think. Let's do that. Okay, there's so many. How dare they live in groups? Okay, do you see any eggs? I feel like I haven't seen any eggs yet, but maybe it's just the wrong season for it.
Yeah, I haven't. I guess if I used Penguin Watch more, I would have an understanding of the seasons.
Yeah.
But because I don't know anything about penguins beyond what they look like.
This one looks smaller to me, so I'm gonna make it a chick.
We could have a look at the subject data to see if it has a timestamp in, and whether the field guide has the seasonal chart of when you're likely to see eggs. Hard to say.
BMIP.
Taken in 2024. I'm gonna hazard a guess.
Really recent.
Yeah.
Or uploaded in 2024.
That's also possible.
Yeah. Now we're just guessing at the research methods.
The importance of metadata.
Yep.
Yeah.
There's also this this fun bit at the bottom. Ultra fire. That might just be the camera or something similar. So let's pan over here. Okay. I think we're back on some adults here. Yep, orange beak. Ooh, that one's completely bent over. Let's call it an adult. And I think we've got the yellow whiskers. That one looks a bit smaller to me, so I'm going to call it a baby. This is a bit arbitrary, but that's kind of the point of citizen science. It would take too long for an individual researcher to do this research if they had to pick out every single one of these. And our computational methods are not good enough yet to be able to do this in an automated way with high enough consistency. So we're reliant on volunteers in order to do a lot of the work. Otherwise, it just would not be possible to do. So even if you are a bit unsure, it's still best to put something down in most cases, because otherwise it would not be possible to represent the data. At the very least, the researchers are getting the locations of each penguin, and then they also are getting some classification information. But like I said, that is the part that is a bit more subjective. So.
And this project has been running for quite a while now, so there are some papers that have been published about this research and about a penguin detection algorithm that they've developed from the outputs from previous data sets.
Yeah. That's fun.
Which I think are probably open access, and certainly like the results updates will be free to read on the Zooniverse webpage of the project.
Oh, wonderful.
But I guess that's another part of these kinds of things, is that you can collect the data with support of volunteers and create perhaps one automated method, but then transferring that to other data sets can be challenging too.
Yeah.
And so it might be that you then need more help to classify slightly different data in order to expand your research.
Exactly, because a lot of times, if a task has been completed in one image, for example, or image type, if it changes at all, like the time of day, the contrast, like a lot of the details of the image can really confuse the computational approach, and make it much harder to be able to continue doing that automated processing. So it is important to always give like little top-ups essentially to the computational approach in order to like make sure that it is performing at its best, and also just to check whether or not it's doing well. Because the other reason to collect from humans is to be able to say, that the algorithm is doing the correct thing compared to what a human might do. Which that also has a little bit of like a fun bias against it though, because if I do this and if Trisha does this, then we might end up disagreeing, and that's totally normal. Like that's a part of the subjectivity of having an opinion and doing a task that isn't straightforward. And so using my markings as like the expert, the correct like outcome, can sometimes create issues then when you are developing an entire tool, like a computational tool based on it. So it is important to kind of keep that in mind as like a researcher in this space, in addition to like using the human output to validate. So.
The other interesting part of that is whether or not how much do you trust the quality of the data from your machine learning output, and how much do you trust the quality from the collective human output? And it's hard to say which of those is more right.
Yes. Yeah. Exactly.
That's very productive. I'm very impressed that you got through all of that.
Thank you. I'm gonna hit next now. Yeah. And hope for a slightly easier one. It did not happen.
They all just stood up.
Yeah, exactly. It's probably like the exact same location. Yeah. Just like 5 minutes later. Oh dear. Okay, I'm gonna do this last one, and then maybe you can spend some time on it.
Yep.
Or we can switch to a new project. Let's find our quadrant.
Do you have a favorite project that you've contributed to on the Zooniverse?
I really liked, there was a trail cam. Oh, what was the animal they were looking for?
It could have been
It could have
anything.
been anything. I
Yeah.
know, yeah. It was something watch, but it wasn't penguins, I think. I can't remember now. It's been a little while. Um,
There's a lot of wild cams.
yeah, there are. There was also one that was focused on tuberculosis that I really enjoyed as well, because I have a background in some biochemical stuff, so it was fun to see the assay that they use and that I had used in the past,
Yeah.
but putting it into the context of like a citizen science project, I enjoyed that.
I haven't tried any projects that have used sound yet
Oh,
because there aren't
yeah.
very many.
No, I agree.
But I think there is one up at the moment.
Mm.
One of the really cool things about the Zooniverse is that you can kind of pick your poison. And maybe you like picking penguins from images, but perhaps instead you like looking at transcribing records. So if you're really good at reading handwriting, there's a project for you.
Oh,
If you just want to listen to like the sound of a mosquito and try and work out what species it is by like how it's buzzing, I think that also has existed in the past. So you can really do anything.
Yeah. The transcription ones sound like my nightmare. Like, I suck at handwriting.
Yeah, some of them are really, really hard,
Mm.
and it's so impressive that there are some volunteers who contribute so much that they start to have an understanding of what they're looking at, and they can pick out stories. I think that's one of the great things about citizen science too, is that you've got fresh eyes looking on something, and what is important to one person
Yeah.
isn't going to be important to someone else. And so actually there's been a lot of really cool discoveries made in different fields, either about you know actually naming the green pea galaxies, right, in Galaxy Zoo,
Yeah.
but also working out that there was a particular person on a particular ship in some kind of muster records, and finding that story and figuring out who that person is. So it goes all the way from kind of that individual human story of history to finding a new type of galaxy, or at least naming a new type of galaxy.
Yeah, no, it is. It's really amazing what's possible with kind of the quote unquote power of citizen science. Because you don't really think like the collective outcome. Like it's not necessarily front of mind when I'm just clicking on penguins, for example. But it really does generate like enough data and enough power that you can really move things forward in interesting ways. So yeah.
I'm wondering if I have a favorite project. I'm not
Yeah.
sure I know. I really
You've
like
you've
the
done a lot
ones
more than I have,
I
so
like to explore. I like to dabble
yeah.
in different projects,
Uh-huh.
and just have a look at what's out there.
Uh-huh.
I don't think I commit really, really heavily to any one project, which is also completely fine.
Yeah.
Um,
Use it however you want.
yeah, one thing I really do enjoy is when you have the project on the app, the Zooniverse app,
Oh yeah.
and you can swipe
Oh yeah. No, those
on your
those
phone.
are those are fun.
Yeah,
'Cause
that
that's just kind of like when you're bored.
yeah.
Like you can just pull it up and do it whenever. And it's amazing how many you can get through in such a short amount of time.
Yeah.
Yeah.
I think it really harnesses the like doom scrolling mechanic of your thumb and uses it for the power of science,
Yeah.
but it's still kind of it's that phone screen time when you're like on the bus or something.
Yeah. Okay. I wasn't as confident about some of these because it's hard to see the beaks.
Yeah.
It really is dependent on which direction the sun is coming from. I don't think I considered this before, but like if the penguin is in shadow, like the beak is in shadow, it becomes hard to see if it's an adult or not.
Yeah.
So,
I think we've picked a particularly different, well, we've been
given.
shown, yeah,
Yeah.
a particularly difficult season perhaps,
Yes.
or a series of photos.
Yeah. Because I think in a lot of these cases, the chicks are quite old
Yeah.
in the juvenile range, so it's a lot harder to distinguish. And I haven't seen any eggs,
No.
which I am a little sad about, but it's okay.
I think they are few and far between, to be fair.
That's true.
Have you seen any other interesting animals?
No, I don't think I have. Oh, you got a different one. Although it's a bit blurry, so good luck.
Thank you.
You're welcome. Magical pen and transfer of power.
Alrighty.
There you go.
Yeah, the question is, do I see brown differently to you?
Oh, okay, yeah.
Yeah. I'm gonna use the zoom in method as you have correctly modeled.
Ooh, that one's like really obvious.
Yeah.
You're getting a good one.
Oh, but some of them are fluffy. I'm going to get through these more slowly than you as well.
Yeah, it's fun to watch like the style of people and as they're doing these tasks, because I find in some cases, perhaps in my case, the decision is just like instantaneous, and then you move on quickly. Whereas
Yeah.
other people really agonize over it, and like go back and forth multiple times, and like especially for things like this where you're putting a point on like an object, like making sure that it's like in the center or like centered or something like that, to like the location of the point relative to the object. I've seen people really like struggle to like allow it to be even a smidge different than what they think it should be. And my experience is that it's generally best practice to like
not care so much.
Not care so much, because like I said, many different people are looking at these things and you're building a consensus. And for the most part, like it should be in most cases, right? It should be fairly obvious from that initial look, so that you can kind of move more quickly. But
yeah, here I would say that like it's questionable whether or not this is an animal or just a blob.
A
but
shadow
if
or something,
it is an animal, it's a chick, and so I'm going to go ahead and pick chick.
Yeah. Okay.
That's one of the other things about looking at this data, is that you can actually zoom in so far that you're going to be choosing between like 2 pixels, and then fretting over exactly the placement is not important at all, because it's the same pixel of the image that you're looking at.
Exactly. Yeah. Like that level of accuracy is unnecessary for the most part, and so it's not really worth the agonizing. So, I agree with you on that one.
Yeah, I think it's got a bit of an afro of chick. Is it fur? Are they feathers? Is there a transition between the two?
I am not the right person to answer that question. Ecology was not my degree.
I studied biology until I was 16 and then I stopped.
Yeah. My biology is more of like the protein level.
Maybe you can answer the question I've had forever, which is, what is a cell?
Oh, I mean it's a bag of liquids and things. Is that sufficient, or?
I mean, if it is the answer, then I will accept it.
I don't know if it will get you like an A+ on your
Mitochondria are the powerhouse of the cell.
There you go. That's it. The answer is always C, kids. Like,
I'm getting some difficult ones where I would say that actually there's a couple of like penguins behind penguins here.
Yeah. I had a couple like that as well, where it was, especially in like the background, it becomes a lot harder to distinguish. So
sometimes it's important to gain context of how far you've zoomed in as well.
Yeah.
I'm gonna go that this one is fluffy enough to be a chick. And I shouldn't really pick that as a chick either. I'm just making an assumption that because there are others like that, then that one is also a chick. And I'm also getting to the point, I think, where these are too far away now.
Yeah.
Maybe I will pick one more adult here, and then... Yeah, not quite got them.
No, that's good.
But I'm done.
Yep.
I've marked 45, so...
That's quite good.
There were too many to count.
Okay. Ooh, we got a sunny day. See, and this is where, like, the direction of the sun, because, like, all of the shadows make it actually quite difficult. Because you see, like, the shadow on the rock and think, is that another penguin, or is that a shadow? In some of the cases, so...
Not the kind of thing that we think about on a day-to-day basis, shadow. We have other imaging problems, don't we?
Yeah, exactly. Which, maybe one of the future sessions of this, we will focus on one of the Science Scribbler projects, and we can talk a little bit more in detail about those. But let's keep to penguins for today.
Penguins for now. Oh, it is hard to tell, isn't it?
It is, yeah.
What do you think about this one?
I was going to say chick.
Yeah, and then adult behind.
Yeah, but you can't see the head of the adult behind because it's doing some like weird. It's like headless almost.
Yeah.
But I think the size difference.
I think that I'm confident that the adult is behind.
Yeah.
And I'm going to put these 2 points really close together, and that's probably going to
confuse
something instead of making it clearer. But that's okay too.
Yeah.
I think I can see whiskers of that one.
It's fun that they call them whiskers because they're like kind of on the very sides of their heads.
I feel like they're kind of like little ear tufts
almost. Yeah, that's what I was thinking too.
See, this is
a confusing
speaking
situation
to me.
with the shadows. Yeah.
Yeah, but I'm gonna avoid it.
Okay. Let's look at, because they haven't actually told us what they're hoping to do here with this. So let's spend a little bit of time looking at their about page. Let me get it up on my phone.
Yeah, why do we care about penguins at all?
Yeah, I mean, they are cute.
Yeah.
But like beyond that. Penguin Watch. So the tagline is count penguin adults, chicks, and eggs in faraway lands to help us understand their lives and environment. Okay. There are 6 people working on this right now, so we are one of 6.
Wow.
And it is 51% complete with the current workflow, so that's pretty good. So let's go to the about page and learn a little bit about it. Okay, so they've been deploying time-lapse cameras to overlook 2 rockhopper penguin colonies on Isla de los Estados in the southern tip of Argentina, so please forgive me.
That was pretty far from here.
Yeah. Southern rockhopper penguins are classified as vulnerable under the Red List of the International Union for Conservation of Nature. It tells us how likely a species is to disappear from the planet, or if it has already. They are hoping to study the breeding behaviors of the southern rockhopper penguins in the context of climate change to better inform conservation management strategies. Okay, so they want to understand things like how successful the penguins are at raising their chicks. So that's one of the reasons they're having us classify into adults and chicks and eggs. And then they want to also identify other individuals like predator birds in the images so that they can learn more about their behavior and interactions with the penguins. They say that the colonies are already declining, and because of that, they're at a higher risk of predation, where other birds, for example, feed on the young and the eggs of the rockhoppers. Okay, so that's from the project that we're working on here. But from one of their other projects, they are doing foraging cameras. So these cameras are set to shoot at 1-minute intervals to capture short behavioral events like parents feeding chicks or changeovers during incubation. Okay, so they didn't have any of that data for us to work on today, so that's interesting though. It really is quite like surprising what you can tell from
a still image.
Still images. Yeah, exactly. That was kind of where I was thinking too. So
I'm gonna say done with this because though there are many remaining, they are all looking away from the camera.
Yep.
So it's pretty hard to hard to say.
I feel like it might be useful for them to have that as a comment, right? Like unclear or something like that. But, you know, in a lot of cases, they don't want you to say unclear, because then everyone would say unclear and you wouldn't get a consensus that's useful. You wouldn't get the classification.
Yeah, 10 people saying I don't know gives no information at all.
Whereas if 10 people say this and another 10 people say that, it tells you there's something there, but it's unclear what it is. So it does the same thing, but in a different manner. Whereas, if 15 people say it's one thing and 5 say another, that's different. So yeah, it is an interesting kind of rock and hard place to be in, but it is best case for the research, I think. So.
Because even having an understanding of which images are challenging, or which areas of whether it's the adults or the chicks that are particularly confusing, can be really important. These guys are relatively...
This guy on the far left is in the position that I assume that I think it's behind our picture. They assume when they're sliding on ice. It just seems like it would be very painful on rock.
On rock, yeah.
Oh dear.
What do you think about this one here? It's hard to say.
That's too blurry.
It's looking brown to me, but it's also looking really blurry.
Yeah, I could see brown and cream, on the front. It's a slightly different. Yeah.
I'm gonna go for it.
Yeah, that's fair enough. You've convinced me.
Yep. Making a choice is important too.
Yeah. I think Alanis Morissette said that.
I think I only know one song, and you know which one it is.
Yeah, of course.
Okay, one more, I think fairly clear.
Okay.
Adult there, and then...
All right.
I'm done.
I think that might be us for today as well.
Yep.
Yeah?
That's enough penguins.
I think so.
Yeah.
This was really fun. I think we got through about 10 images, and each was probably, we had a lot of penguins in them.
So. Yeah, I think 30 to 50 penguins per image.
In most of them, yeah. So, and that's all you need to do. It's not, onerous, just a couple a day, and you can really see a lot of improvement. So, all right, well, thanks for tuning in with us for our first ever stream, and I'm sure we'll be back soon. We'll pick another project and we'll do it again.